hearing anyone objecting to having the recording on. I've just started it, but like I said, we can pause it at any stage. This is not something that you have to do if you're not finding it comfortable to speak while there's a recording going on. Like I said, I always try to block out, and if there's something that discussion goes to personal things or student needs, I totally drop that out from the recording that I make available for the rest of the class. But I think it's kind of nice for the others to have that as well. So what I was going to propose that we do today, we'll do kind of like even more condensed version from Wednesday's session. And I do want to also, like I said, I'll do an announcement, but I'll do want to also do it on a recording, that question 2 on the quiz 12. Because we talked before the recording started about that, because I don't think that that question is super clear. So first of all, happy 4th of July if you're celebrating. And I didn't necessarily do the greatest job when planning these sessions. I would not expect anyone to spend their 4th of July here, but since you are here, I really do appreciate it. Trust me, I would emphasize that if you ever need letters of reference or otherwise that you're going above and beyond to attend to every single session. That is not required. You've already done, all 3 of you on this call, more than you have to do to get that A in this class. I also wanted to kind of share quickly, I don't think that it's really a concern for most students, but because sometimes I do get students and I'm pretty responsive when they reach out to me, and then if there's a longer of a wait, say 24 hours, they get really confused. Why is he not answering to my messages, or why is he not grading things? I have had a family member going through surgery earlier this week, and I've been spending a lot of time at the hospital as a result of that. And as much as I tried, I didn't really want to connect our course materials to a public network just for the data safety and safety of all of the potential things that could have gone wrong. So if you have been waiting for a response from me, or if you haven't had your assignment graded, I will literally do it after this session. But I'm just kind of sharing where I'm at. And having said that, I want to extend the same grace towards each and every one of you. If you ever are behind on assignments, if you ever need extensions and so on, let me know. We'll figure it out. There are some limits to that, though. I'm going to be honest, my salary ends on the last day of our class, so I can't see myself really being super eager of reopening assignments and working on this course after the semester has officially ended. I don't think that that's going to happen. But up till then, if you've missed anything, if you want to reattempt something, if you need something reopened, I'll deal with those on a case-by-case basis. So don't feel like you can't reach out and ask. The worst thing that could always happen is that someone says no, and that's not a big deal. Also, I did want to recognize that we are right at the finish line on this course. I do have a tendency, and I've shared this before we started recording, I have a tendency of building my courses a little bit heavier towards the first half of the course, and then the second half of the course is always a little lighter. That's because things happen. Things happen in students' life, things happen, we all remember, maybe we try not to remember, pandemic and so on. So just having learned from those experiences, I think it's always useful to have that little bit of a time towards end of the semester when we can kind of catch up on anything that might have gotten missed. Or if anyone is wanting to work ahead on the course, you totally can do so, and you can wrap up the course early. There is a good chance that I won't be grading the manually graded assignments or discussions ahead of the time, but once we get to that week, I will totally do those. But if you know that you have things to do, places to be, and you want to work ahead, you totally can. But we're reaching the final hurdle on this course. And having said that, I think that what we're seeing on this screen is a good reminder of that we've covered ton of material. And I feel like my students often say on 201 that we've covered a ton of material that's looking at little things on little areas. I do work really hard trying to build kind of a narrative through the chapters that we look at. You saw that we started with some basics, and then we kind of reminded ourselves of the concepts that you should know, and then we started building up from that. We really use the integumentary system because it provides, again, kind of a review of main tissue types, but then looking at how bone, cartilages, skeleton, muscles are all connected. This final third of the course, I guess, is looking at nervous system. So we'll be focusing on nervous system through various different chapters, and we'll be looking at it from a little bit of different angles. You'll probably notice that the amount of homework is not as high. I think last week was, and I'm taking that on myself, that last week was probably a little heavier than what's fair towards the students. So I'll look that for next semester. But having said that as well, I don't think that there's going to be as heavy workload, but we'll look at nervous system from various different perspectives. And on this week, we've kind of done an introduction to nervous system and really focused on the central nervous system, which is the part that students often think when we talk about nervous system. So thinking about the brain, thinking about spinal cord, but we'll cover the rest of the nervous system as well in the coming weeks. But I think, like I said, you'll notice that the workload is a little lighter than it was at the early part of the semester. And then you just have one more set of tests. You have your exam tree and practical tree. I don't have a cumulative final exam on this course because summer is so short. So you have done everything you need to know about the previous chapters, and only things that you'll be tested for the rest of this semester is going to be nervous system. So I did kind of share this, and I think today is the last day, if I don't remember wrongly, but I asked that if anyone wants to submit some feedback to me. I try to offer many ways of letting me know what works, what doesn't work, and I also try to offer ways of letting me know while the semester is going on, so that we can do changes and we can do adjustments. So there is that survey. I did have at least a few students who were a little confused that, well, where do I find it? If you click on that link where it says complete midterm feedback here on your learning modules, that will throw you to the actual survey. I have shared the same link also on announcements. This is a little different survey. Sometimes students are expecting that the survey link gets emailed to you. That's not the case this time around. The links that get emailed to you for surveys are kind of what we call our official end of semester surveys, and we do not conduct those during the summer. Not my choice, it's the institution's choice. But I think that just as much as I want to hear feedback from my students during fall and spring, I do want to hear from my students. So I do independently the survey. The results of the survey, however, do go to my boss, and they do go to the board that reviews my contract every year for the first 5 years that you're employed at our institution. So I probably wouldn't write swear words or things like that to that. I do also give a little bit of extra credit for anyone who has completed that survey, but I also want the survey to be anonymous. So what I ask is that once you've filled in the survey, grab a screenshot of the final page or use your phone to take a picture of that screen where it says that you've done a survey, and upload that to that assignment area that you'll see at the bottom of that feedback module. If you forget to take a picture, you can just literally type in to a text box on that area that, hey, I completed the survey on this day and approximately at that time. Like I said, I treat those anonymously. I will not match it. I won't be able to match it. The survey doesn't record who submitted, who said what. So that's an option. Again, no requirement to do that, but if that's something that you think that is worthwhile to share with me, what's working well, and also where can I do better, or where can we do differently, I do appreciate I'm not going to go through really of these kind of feel good and reflective activities. We started doing those a little bit more after the pandemic and during the pandemic, but there is a nice quote that kind of came up for this week about looking inwards and seeing a universe inside you. And I think that that's really what nervous system is. Nervous system is basically giving meaning to all of the information that your sensory organs collect. So it is good and nice to have sensory organs. It is good and nice to have also effectors in our body, whether it's muscles, whether it's glands, or otherwise how we interact with the world. But really that meaning for all of those happens at the level of nervous system. And what I think is interesting is that all of us perceive the world around us and world inside us and how we interact with the world as well differently than everyone else. So there's a certain level of subjectivity, even though we're still able to live in a society where we share values, we share, for example, what's acceptable, what's not, we share aesthetic views or ideas, we share what's good and proper and what's not. Those are all creations, and they're creations where we've just ended up coming to a conclusion that, hey, I see this, you see this, let's set this as a standard. So nervous system is interesting. Having said that as well, nervous system is incredibly complex. One of the things that I do outside teaching is that I do study law, and I do study also ethics, and not in the interest of practicing, but we see a lot of more challenging medical cases that focus on people whose nervous system might be in states that are not clearly that things are working perfectly or things are not working at all, but we're somewhere in the middle on that boundary. So I'm talking about things like persistent vegetative state, I'm talking about things like mental health disorders or even substance abuse disorders. Those become a little bit more challenging for healthcare professionals, and they become a little bit more challenging also when it comes to the law that we have to honor. We kind of highlight on our Western medicine and medicine in US, especially the patient autonomy. But there are times when patient autonomy no longer takes the number one step. For example, if patient autonomy would mean that the patient would be harming themselves, or they would be making choices that would be severely damaging to themselves. And if we start to interfere with that autonomy on those stages, then we have to look at that. Is the patient who's making those decisions capable of making decisions? If they are capable, you do have a right to make bad choices. You do have a right to make even choices that are medically harmful for yourself, but you must have capacity. And capacity assessments are something that is reserved for only certain fields of medicine. And typically we tend to have at least 2 independent practitioners who do that assessment. So that's where I'm interested. And the reason why I wanted to share about is that so much of that has to do with the nervous system, where we have to look at those things. I always emphasize to my students also in medical school that addictions are medical conditions where the individual doesn't necessarily have full capacity. And if you don't have full capacity, you might not be able to exercise your autonomy the way that you should be. So then we need to start looking, like I said, other values like beneficence and non-maleficence and so on in terms of how do we care for the patient. That went a little dark and a little gloomy, but an interesting field. I also use this slide occasionally just to show, again, that driver in that point that your nervous system, and especially your central nervous system, is where everything that you experience, everything that you have experienced, memories of that, and your dreams and hopes of everything that you hope to experience takes place. Everything else in your body is just kind of protective and support structures for the nervous system to be able to do that. I don't know if I totally believe in that statement, but I do think that it's an interesting way of looking at nervous system and importance of that. I think it was less than a year ago, there was this big, at least I don't know if it showed up in mainstream media, but on my field, it showed up a lot that people are now starting to look at if eternal life can be accomplished rather than trying to keep the whole body alive by keeping individuals' brain alive indefinitely. And there are some discussions, and I know there's also some experiments that have been conducted that could we just salvage the brain of an individual whose body is already starting to fail due to the old age. We do all have a limit to how long we can live physically. In terms of our biology, there are some certain boundaries and certain limits, even though we keep pushing them further. But maybe one way how future looks like is that we're focusing mainly on the central nervous system and preserving that. So I don't know. That's a kind of an abstract concept. Here's the assignments for this week. Before Sunday midnight, I'm asking you to do quiz, practice anatomy, and discussion forum for chapters 12 and 14. I think that 14, 13 chapters, I pick and choose sections of those for this week, and then the rest is going to be next week. So don't feel like you have to master every single nuance of those. But there are all kinds of materials from those chapters. For discussions, I'm looking for 2 discussion posts per discussion forum for full marks. You can always do more, and I always appreciate if you do more, but there is no expectation of that. And do remember that you don't have to do everything. You just need to do enough to get that A if you're chasing for an A, or if you're chasing for a B, you need to do enough to get 81% on this course. Or if you're happy with a C, you have to just get 71% on this course. So however you collect those points, that's up to you, but that's why I offer those extra credits as well. I did want to, again, and I know that we've already talked about this, but I tried to talk about it in multiple settings so everyone hears it. I just wanted to share that next week, not only that this week I have been a little bit distracted with family healthcare issues, but next week I do have a situation that I'm sent by my employer to a work trip. And the work trip is at the Howard Hodge Medical Institute's campus in Virginia. As a result, there is limited internet accessibility, not because there's no accessibility there, but there's some research that is kind of limited that might not be shared at all times. So I might not have internet available all of the time during that trip. The schedule for the trip is still kind of changing a little bit. And just to be cautious, what I have done is that I've said that we're not going to have our live online sessions next week. It is possible that I'm available, and if I'm available, I will let you know through Canvas messages, but it would be likely that the time would be different than when we normally meet. I will, however, record a session like we would have now so that you have something fresh and something new to look at. But if I take longer to respond to messages, if I take longer to grade assignments, that's part of that. I will be also taking a little bit of a step back. It's not just because of the trip, it's actually part of how I kind of built this course, that we have students taking more and more ownership of the discussion forums as the semester continues. You might remember the first couple of weeks, it was literally just one post that you needed to do. But the expectation is kind of growing as the semester goes on. And I'm probably going to be less present on those forums. Next week, it's going to be more of students presenting questions and students answering to questions. But all of that I will share at the beginning of the week through an announcement. So that leads us to the material regarding to the chapter 12. And one of the books, if I have students who always ask, what else could I read? And what's not necessarily so much of a school book, but an entertaining book. One of the books that I encourage my students, if they want to read something a little lighter, is a book called The Man Who Mistook His Wife for a Hat, written by a famous neurologist, Oliver Sacks. Oliver Sacks outlines on that book. It is a little older book, but he outlines on that book some of the curious cases that he has worked with. And one of these was Dr. P, who had visual agnosia. So nothing wrong with vision or visual organs or even intelligence. But where the error happened is that somewhere along the brain, where he was giving meanings to what he sees, there was a disruption. That's what visual agnosia is. It's a impaired recognition of visually presented objects. So when he looked at his wife, he actually saw a hat. And that obviously caused some tensions. He tried to put the hat on the hat shelf, where one would probably normally do that, but in reality it was a wife, and wife obviously didn't like that too much. So interesting case scenarios if you're looking for something fun to read. If you can find the book, I have many copies in my office. I'm happy to loan you one. Let me know and we'll work through that. The other case that I share normally with my students is regarding Phineas Gage, who was an American railroad construction foreman. And when he was working on blowing up some of the rock material out of the way of the railroad at that era, it was done by drilling holes to the rock and filling it with black powder manually and then exploding it to kind of remove that material. Well, he was doing manually that work, and he was using a metal rod to push the black powder into that hole, and the metal hitting with the rock created a spark, and as a result the black powder exploded, and the metal rod ended up penetrating through his skull and especially impacting his frontal lobe. And we see from cases like this and other cases that when we have damages to certain areas of the brain, we see that certain functions are changed, and that's how we've been able to get a lot of information about one's brain, one's central nervous system, what functions are mainly focused on certain areas of the brain. Having said that, I will recognize that brain is what we describe as highly plastic. So it's able to reallocate functions from other areas where they normally would be housed to new areas if necessary. I think one good example of that we see is with mice studies on mice when they have their whiskers. Each whisker has a specific brain area that's dedicated for that. But if now for some reason that brain area would get damaged, the whisker information would be allocated to a different brain area. So we see that there's some amount of being able to be responsive. But back to the Phineas Gage, after his injury, he lived for another over 12 years, and people said that his personality and his behavior changed. He was in the past very calm and relaxed man. He turned into very irritable, kind of a mean, kind of an unpleasant guy. So again, we see that these injuries have been a major source of information. We these days rely much more heavily on functional brain imaging, which is a form. For example, if you're interested in medical imaging, not only do we do MRI, which looks at the structures, but we also do what's known as functional MRI, and there we look at the blood flow and slightest of differences to different areas of brain, which allows us then to associate that when a part of a brain is super active when doing a certain type of activity, we can associate those processes to those parts of the brain. So those are kind of giving some background. In terms of the nervous system is one of our two bodies communication systems. Nervous system is what we'll focus on this course, and then BIO202 you'll start by talking about endocrine system, which is all about hormones. So nervous system, electrical impulses and chemical impulses play a role in nervous system. So we see that you can consider a lot of your nerves almost like as electric wires passing messages from one body part to another body part, but that it's not completely just electrical messages wherever that neuron ends and the message moves to a next neuron or message moves to a target organ. That short distance of a message takes place through neurotransmitters. So it's chemical message. Messages in nervous system are very fast. They're very specific. They start from somewhere, they end for somewhere, they travel within less than a second. So we're talking about milliseconds in terms of the timeline. Whereas when we get on BIO202 to look at endocrine system, we see that hormones, they can take a long time for messages to pass. Certain hormones are quick, but certain hormones, it can take up to months before we see those effects. Hormones travel throughout your entire body, and they also tend to be longer lasting, whereas in nervous system, once a nerve impulse has passed, it's gone. In endocrine system, hormones tend to linger around a lot of the time. So that's something kind of to preface what's going to come up in your future courses and what we're looking at in this setting. I also wanted to show the three functions of the nervous system. There's many ways of classifying nervous system and functions of nervous system, but I do find this as rather useful. We see that your nervous system is responsible for collecting sensory inputs, so gathering information through your sensory organs. This could be many, both internal and external state of the body that we're looking at, whether it's smell, taste, sight, hearing, balance, touch, position, or otherwise. So we're collecting information about the world around us and within us. We also integrate that information, so we process and we give meanings to that information. So that's really where we see that central nervous system is crucial. And then motor output, not always and not for everything, but for certain things, your nervous system decides that, yes, we're going to act on this. So we recruit our effectors. Typically, these are muscles and glands to interact with the world around us. Do remember that we are constantly gathering more sensory input than we could potentially process in our brain. So there's also that selection that takes place, and we'll talk about when we look at specific parts of the central nervous system, which are kind of relay stations and kind of stations where we choose what comes to the consciousness in terms of that as well. I kind of love that diagram that I showed earlier because it fits in very nicely with the concept of homeostasis, which is something that we've been talking about in the past. Homeostasis is such a crucial concept. We'll use that to explain many things in the body. So we'll see that you could kind of fit in the three functions of the nervous system to this homeostatic feedback mechanism model as well. We can divide nervous system in many different ways. Probably the most common way to do this division is to divide nervous system into central nervous system and peripheral nervous system. Your central nervous system includes your brain and your spinal cord. You can consider this kind of as the computing or processing part of the nervous system. So a lot of integration and a lot of control. Interestingly, these both structures are located at the dorsal side of the body. So in your cranial cavity and your spinal cavity, where the spinal cord runs. Peripheral nervous system instead is all of the structures of the nervous system outside the CNS. So nerves and sensory organs, you can consider a lot of those as communication lines or receivers or antennas, however you wish to frame it. They are especially what we'll be looking at. We'll be looking at your spinal nerves and your cranial nerves, but also, like I said, your special sensory organs would play a role in this. We can actually divide our peripheral nervous system even further. Peripheral nervous system can be divided into somatic and autonomic branches. Somatic branch is what's under voluntary control, controlling things like movements and actions, whereas autonomic branch is involuntary. And involuntary or autonomic branch is responsible for our parasympathetic and sympathetic branches. So sympathetic branch is fight or flight responses, parasympathetic branch, your rest and digest responses. So based on what your body is doing, is it dealing with a crisis, with a risk for your well-being or your safety or your health? That's when we would activate our sympathetic nervous system, which is really stress responses, whether it's stress and excitement or fear or anger or embarrassment even. We see that things like your heart rate go up, your blood pressure goes up, your respiratory system structures literally dilate so we can get more air to the lungs. We see your blood vessels as well dilate, especially the blood vessels that are directing blood to skeletal muscles. So we can literally fight or escape a threatening situation. Instead, when your parasympathetic nervous system is active, we really see that most body processes get depressed. We don't really need to have those running at the fast rate. Only processes where they are kind of elevated would be your digestive system, your urinary system as well. So we're kind of doing body's maintenance functions, digesting food, extracting nutrients and energy from the food, and also producing our waste products like urine. So kind of two branches of the autonomic nervous system. I jumped over this division that nervous system can be also divided into sensory or motor branches. I think we touched upon that already. Sensory branch is kind of described as an afferent, motor as efferent. This is in relation to the messages and central nervous system. So afferent messages are messages that are coming into the central nervous system, whereas efferent are messages that are leaving central nervous system. What I normally like to talk next with my students is the different parts of a neuron. And rather than drawing it, I'm going to pull up a previous sketch that I have. Just give me a second. I'll pull that up. I think it's going to be a kind of a quicker way to look at some of the important structures. That, and I'm going to do also the next one. So I'm going to reshare my screen. You should be able to see the screen now. So in terms of neurons, I want to be again transparent and say that there's a lot of variability in neurons. Neurons are, in terms of their anatomy, again, complementary to their function. And we see many, many, many different arrangements. So what we do, rather than making students at this level to learn many different kinds of neurons, we show students what's kind of described as a standard neuron. In fact, you would probably not see a neuron like this in the body. So this is a schematic representation, oversimplification of what a neuron looks like, but it captures all of the key parts. So let's have a look at the parts of a neuron. So we have dendrites. Dendrites are responsible for collecting information that travels into the neuron's cell body and nucleus. And within that cell body and nucleus is where we kind of do summation of all of the information coming in through dendrites to make that assessment. Is it enough to trigger a response from this neuron? Or does it stay below that threshold that triggers a response? And that's where we see the action potential. So neuron is constantly assessing, is it worthwhile to create an action potential? Well, if a neuron deems that an action potential is appropriate, we have a portion that leaves the cell body of the neuron known as axon. It leaves the cell body of a neuron at the axon hillock. And this axon is kind of like a long branch. But in this long branch, messages can only travel to one direction. And that direction is away from the cell body towards the axon terminal. Some axons separate into multiple branches, and all of those branches would have their own axon terminals. Now, once the electrical message has propagated through the axon to the action terminals, at the action terminal, we'll see that it doesn't connect directly to the next structure. So the next structure could be another neuron, or it could be an organ that we are trying to impact with the neuron's action potential. So in that case, there's a small gap between the neuron and the next structure. And this is where we release those chemical messengers that are known as neurotransmitters. So far, the messages in the nervous system that we've been looking at have been electrical, but in this space, it's going to be chemical messages. These other branches of the axon, I didn't mention that, they are known as collateral branches. You can also see sometimes that axon is surrounded by myelin, kind of blobs of fatty tissue. And myelin helps us in two different ways. It isolates axons, but it also helps for axons to travel or if the message is within the axon, to travel quicker. So it speeds up the propagation of those electrical signals within the neurons. So those were some of the key parts of the neurons that I would like you to know. Again, you will end up seeing that in some neurons, dendrites take a large portion, and then the cell body might be further along in halfway through the entire size of a neuron. Or it might be that we have axon on both sides of the cell body, or it might be that we have multiple branches, or it might be that there's no dendrites, or there's just one dendrite. So many, many, many different arrangements, but if you know these basic parts, you're able to apply those to specific neurons that you'll see. And what I find interesting is that for so long, much of our research on nervous system has been focusing on understanding neurons. Whereas in reality, just as important structure as a neuron, a single nervous cell, is in the nervous system is what's known as neuroglia or glial cells. Glial cells are supporting cells, so they're not neurons themselves, but they assist neurons. And how they assist, it can be physiologically supplying resources, nutrients, energy, oxygen, removing waste products, or it can be literally holding the neuron on the shape that or place that it needs to be. And there's different kinds of glial cells. The first ones that we see on this illustration are our astrocytes. Astrocytes receive their name because they are star-shaped. And they play a particularly important role in metabolism. They also do produce myelin. Then we have our oligodendrocytes. Oligodendrocytes, their main function is to produce myelin that then wraps around those axons. We also have microglia. Microglia is very, very small, often described as looking like spider-like, and they're phagocytotic cells, so they dispose material, get rid of waste products, for example. And finally, we have our ependyma or ependymal cells. These are glial cells that typically line cavities. Their appearance is that they typically have cilia at one end of theirs. We'll see ependymal cells, for example, in your spinal canal and in ventricles of the brain, which we'll be looking at in a second. But what I was saying is that glial cells have been overlooked in a lot of the research. So we end up seeing that glial cells are actually reason for some pathologies that we previously thought that were related to the neurons. So for example, Alzheimer's disease, a lot of the research interest has moved away from neurons to looking at the role of glial cells in those. What else could we say? We could talk about action potential, which is what we started before the recording started, by talking about that a little bit. And halfway through this, I'm going to jump into that question that I wanted to talk a little bit about. But let's talk about action potential. So what we see at the bottom of the screen is a neuron. We just went through the main parts of that. And what has been highlighted is the axon of a neuron. So that long pathway where the messages traveled away from the cell body towards the axon terminal. And when this electrical message passes here, we can actually measure it. So we see that the insides of the neuron are typically charged at -70 millivolts. And when an action potential comes, so this electrical signal passes through this axon, the negative charge actually neutralizes. It can become slightly positive, and then it returns back to negative. So there's a momentarily positive message passing along the axon. And how we can measure that is by actually taking a very, very fine tip of an electrode that we stuck into that axon. We don't want to go through it. We want the tip to be inside an axon. So that's a skill to do it. Typically, that microelectrodes are made of glass that's heated under a flame so that they become very thin, very sharp, and we can put an electrode inside the axon. We also need to have a ground electron because we're always comparing two charges. That ground electron is somewhere outside the neuron. And we see that there was that negative charge inside the neuron in comparison to the positive charge outside the neuron. Remember, charges are always proportional or relational, so you have to compare something to something else to tell their relationship in terms of the charges. Well, now we have this negative charge inside the neuron, and the action potential propagated along the axon as a change away from this negative charge. So let's have a look at that a little bit. And this is what I would say that most action potentials look like. So action potentials don't actually start until that signal is sent by the neuron and neuron cell body, where we do that determination. So normally the cell was in a resting potential, which was negative 70 millivolts. And then there's some kind of a stimulus. In this case that we've been discussing, it could be, for example, that we're receiving information through dendrites, and then we decide that there's enough information to justify for a message to be triggered. Well, when action potential is triggered, the charges inside the neuron first become neutralized and then shoot above that and become positive. And this is because during the resting potential, we had our sodium-potassium pump and also our calcium, which we saw earlier on that was everywhere, but especially that sodium-potassium pump constantly maintaining that resting potential. And how we maintain the negative resting potential is that for every sodium that we pump out, so we pump out 3 sodiums out from the neuron, we pump in 2 potassium charges or 2 potassium molecules. So in proportion, we get rid of pluses from inside the neuron, and we end up the inside of the neuron becoming negatively charged in comparison to the outside of the neuron. So that's how we established that negative 70. And now the stimulus took place, and we see that the sodium channels open up and sodium rushes into the cell, and that causes the cell's charge to neutralize and become slightly positive, just momentarily though. This stage is known as depolarization. So remember, polar means that you're far away from 2 ends. So depolarization is that we come close, and it's referring we come close to 0. We do overshoot, actually, so we go to that positive charge. We can't stay in that stage forever. So as soon as depolarization ends, we move on to a stage known as repolarization. During repolarization, your sodium channels close and your potassium channels open. So we see potassium rushing out of the neuron and the neuron's internal charge becoming again negative. What's interesting is that we often go slightly below that negative 70 millivolts inside the neuron. And this is important. This hyperpolarization is what gives us what's known as our refractory period. Refractory period is the period of time during which another action potential cannot be triggered. So neurons cannot fire constantly messages. They have to take a slight break every once in a while. And it is during this hyperpolarization that we're taking that break so that we don't end up with dysfunction of a nervous system, for example. And then we return back to the resting potential. I didn't get to speak about that last time in our live online session on Wednesday, but a good example of when action potentials and firing of them goes wrong or goes rogue is when the individual has epilepsy. Epilepsy starts always in a particular part of the brain, and then it spreads uncontrollably from there throughout the entire brain. And the part that connects these 2 cerebral hemispheres is known as corpus callosum. And I think I have a brain model. Give me a second. I'll try to pick it up. Sorry about that. I knew that I had a brain model somewhere, and I do indeed have. So if we look inside the brain, we'll see that the part that's kind of a wide, large band that connects the 2 cerebral hemispheres, that was our corpus callosum. And the function of corpus callosum is really to pass messages from one part of the brain to the other part of the brain. And in severe cases of epilepsy, where now that unsynchronized firing of action potentials is firing everywhere, we might need to actually cut the corpus callosum. When we cut the corpus callosum, epilepsy cannot spread throughout the brain uncontrollably anymore. The problem is that if we do that, we also cut all of the positive and necessary messages from passing from one part of the brain to another. So this is really our last option that we typically resort to. We'd rather not do that. This is actually very much how back in the olden days, when they still did do procedure known as lobotomy, where we basically took individuals who had major issues, major behavioral issues, they were a danger to the society or otherwise, and we basically tried to kill their nervous system, central nervous system, so they wouldn't be doing those horrible things, even in a care setting. What we did sneak in a knife and cut some of the frontal lobe, but especially try to cut those connections of the brain from another area. And when you destroy frontal lobe, like we'll see in just a second, you actually destroy the individual's ability to have kind of things like executive functions, planning, things like that. So yes, it gets rid of some of the more severe behavioral and criminal tendencies, but it probably also destroys rest of that individual. So these days, I don't think that that would be allowed anymore. So that was our action potential. We obviously, kind of going back to what I said about epilepsy, we obviously these days have great medications that we can use to restrict that uncontrollable firing. So surgery is incredibly rare, but it does still sometimes become something that's necessary. So far we have been looking at an action potential traveling along the axon. And remember, the charge changes momentarily in each part of the action. It doesn't change like all action is positive. It's just like a spark that travels through the axon. So that's what this diagram on the side is trying to illustrate, that message passes along the axon. Well, if we have myelin around the axon, what happens is that this message is able to jump from one myelin cell gap to the next gap. So myelin allows us to speed up the propagation of action potentials within the axon. So it provides us a kind of a bypass that we can skip all these steps that fall within the myelin, and we just go from one gap to another, so we get much quicker propagation of messages along the axon. Here we see an actual image of a synapsis. So synapsis was the ending of a neuron. And we see that it could be in connection to a next neuron. In that case, we would talk about presynaptic neuron and postsynaptic neuron, or it could be a neuron and the target organ, like we saw in neuromuscular junction that we talked about earlier. What we see is that the synapsis was the gap between these two structures. There's literally a space where the electrical signal cannot pass through, and that's where we release the neurotransmitters that pass that message in a chemical format. So here I've just kind of explained the same concept, and we see an illustration. So what we see is that an action potential is traveling down the neuron to the action terminal, where we house within vesicles chemicals. These chemicals are neurotransmitters, and these chemicals are now released into the synaptic gap. And once they reach the receptors on the other side of the synapses, they trigger a response, and typically that could be, for example, a next action potential being triggered. Some of the neurotransmitters are taken back into the axon terminal and can be reused. Some of them are deactivated, and some of them are broken down and eliminated. So the neurotransmitters over time decrease from that synaptic space. The parts we had are presynaptic neuron and postsynaptic neuron. The neurotransmitter needs to have a particular receptor for it. So I often describe it to my students like a lock and key mechanism. So not every neurotransmitter will activate every receptor. They need to be a right fit. Similarly, as I have key, I can't open every lock in everyone's houses. It opens only the locks that it's meant to open. So neurotransmitters are incredibly carefully designed to fit into only certain receptors. And that causes some issues, as you'll see in BIO202, when we talk about things like endocrine disruptors, where we have man-made chemicals that mimic natural hormones or neurotransmitters, and we accidentally end up activating structures that we shouldn't. That concludes the chapter 12. Regarding chapter 14, we're looking at central nervous system. So we're really focusing on the brain. I don't think we talk too much about the spinal cord, but remember that nearly all animals have some kind of a nervous system. Whether you're an earthworm or a highly complex animal like dolphin, we do have some reason to believe that dolphins might actually have even more sophisticated brain, central nervous system than humans. Dolphins, for example, never fully sleep. They only sleep with half of their brain. So other half is active, so they're able to still remain able to function, but they just rest half of their central nervous system at the time. But all animals have some kind of nervous system. I guess that single cell bacteria probably wouldn't have, but any multicellular animal, we do tend to see some adaptation of a nervous system. Well, humans and our evolutionary ancestors, like other animals, have highly complex central nervous systems, so highly complex brains. If we compare the brains and the structures of the brains from humans to our evolutionary cousins, relatives, we see that there are certain parts that are repeated. For example, your visual area, your motor control area, and your somatosensory area. They are represented in all of the animals. However, the proportions of these areas depend on what that animal relies on for their survival. For example, we see that sheep don't have terribly good executive functions, so their frontal lobe is very small, but they have good visual processing. They have good smell. For example, cats and dogs have good sense of smell, so we see that those regions are far larger than in the humans. So you'll be able to see on this chapter's materials, I also have a video of dissection of a sheep brain. So you'll see some similarities with human brain, but also some differences with the human brain. So talking in terms of evolution, we're going from the past to the present modern human. We see that one of the things that has allowed humans to end up being what we are and do what we do is that the size of the brain grew drastically. And especially the size of the frontal lobe of the brain grew drastically. This is because frontal lobe is considered as evolutionary later appearance to our central nervous system structures. And it is the part where we house a lot of things that make modern humans capable of doing everything that we do. In addition, not only the size of the brain grew, but the size of the cortex, so the outermost layer of the brain grew. And as we tried to fit in more cortex, one of the ways to fit in more was to start to have these folds, these ridges and depressions on the surface of the brain, so we could fit in a lot of surface area there. So that's something to keep on mind as well. Here we see, going from our early evolutionary relatives, not only the size of the frontal lobe that has grown in comparison to our early ancestors, but like I said, that folding appearance of the brain as well. In terms of your development, we'll see that your brain, baby grows at the fast rate and brain grows at the fast rate in the body, both before your birth and after your birth, until you get to about your maybe 2-3 years of age. the growth rate starts to slow down. And then once you get to about 5 or 6, we don't see huge increase in the size of the brain at the similar rate. But remember, the brain keeps reorganizing all the time. That is what's happening when you're learning. You're creating new connections with the neurons, new synapses are formed, and old synapses are broken apart. So that's why taking care of your brain is so important. If you do use, for example, recreational drugs, alcohol, medications, other things, you are basically altering your brain and altering the functioning of your brain. And sometimes those alterations are permanent. So I would encourage you, since we know that we don't really produce a lot of new neurons, there's a slight amount of new neuron production, but really that's very, very small amount. For that reason, we want to take care of all of the neurons that we have in our body and not destroy them, especially at the brain. Like we discussed, the brain developed at a quite quick rate. Your nervous system starts as what's known as a neural tube, so tube-like structure during that early development after fertilization. Soon it starts to expand at one end and have these areas that expand and fold. And this is what will become your brain. So in this illustration, we see an individual's brain illustrated once they're at 5 weeks from fertilization, so very early in the development. Then at 13 weeks, we already start to see parts of the brain that will be present at the final version. And then at the birth, we see pretty much fully formed brain in terms of the regions that we'll see there. We will look at 4 different parts of the brain. We'll start by talking about the cerebral hemispheres. Those are these big parts of the brain that we often see outside. We will also see inside the brain a part known as diencephalon. So this middle part, and let me see if I can get it to come out. It's been a while since I've used this brain, so it's a little sticky, but I think we can do that. So here we have our diencephalon that we can see, the kind of hidden inside the cerebral hemispheres. And from the diencephalon, we also see our brain stem with the specific adaptations and specific parts that we'll be talking about. In addition, let's see, I might come to regret that I took the brain apart. Let's see if I can get this back together. Just a second. I think this is working out. I'm just trying to show you another part that's going to be important. We also see a part that sits underneath on the posterior side of our cerebral hemispheres. And this is going to be our cerebellum. So this is what's often known as a little brain. And that's particularly important when we think of motor coordination. I sometimes have dancers in my class, for example, ballet dancers. They would have highly developed cerebellum so they can coordinate their body, their motor functions, kind of synchronize those. Another thing that's interesting and worthwhile to note, if we take a cut through the brain, so let's say that we take a coronal plane through the brain that you can see on the screen, we can see that the outside of the brain, which was known as cortex, is made of gray matter. This gray matter is where the cell bodies of neurons are housed, and this is really where the processing of the information takes place. Inside of the brain is mainly white matter, which is myelinated axons passing messages from one part of the gray matter to another part. We will also see that there are specific blobs, if you wish, inside the brain, and these are nuclei that we'll be talking more about later on. They are going to be specific areas that are responsible for specific functions or specific processing. We'll also see empty spaces inside the brain. And all of these empty spaces connect when we look at them three-dimensionally. And these are what are known as our ventricles of the brain. They're filled with a liquid that we'll talk about in just a second, CSF. The other thing that I wanted to show in terms of our spinal cord, we have there as well gray matter and white matter, but now the arrangement has been flipped around, that white matter is more outer and gray matter is more inner. And we'll talk about spinal cord and the processing of information on there. So not all information is processed at the brain, some also at the spinal cord. We'll talk about that in the coming chapters. So let's have a look at those ventricles, those empty spaces inside the brain. Like I said, they're filled with cerebrospinal fluid. Cerebrospinal fluid serves kind of two different important purposes. It helps to pass materials, nutrients, it helps to remove waste products. It helps to pass also hormones. So really kind of facilitating that transportation within the brain. There is vasculature in the brain, important blood vessels that you'll learn about as well, but these ventricles are important for us. Remember that cerebrospinal fluid also bathes the brain from around it, so from the outer side. Your brain is kind of like sitting in a, if you wish to imagine your skull as a bucket, in a bucket of liquid, and that kind of protects that you don't get damage to the surface of the brain, for example, as easily when whiplash or otherwise would take place. So let's have a look of each of those four parts that we promised to address, and let's start with cerebral hemispheres. Cerebral hemispheres represent about 83% of the entire brain mass. Some of the terms that we want to know when we talk about cerebral hemispheres, we have these ridges or bumps. These are known as gyri. We have these grooves or depressions. These are known as sulci. And when you have very, very, very deep grooves or depressions, they are what we know as fissures. And these are important terms because even though we all differ in the surface texture of our cerebral hemispheres, there's some common structures that we see in all individuals. And first of these is going to be a longitudinal fissure. Longitudinal fissure separates your brain into right and left hemispheres. So it runs longitudinally throughout, creating your right and left side of the brain. So that was our longitudinal fissure. Another fissure that might be worthwhile to notice is what's known as our transverse cerebral fissure that separates the little brain, if you wish, from the cerebral hemispheres. So cerebrum and cerebellum are separated with this fissure. Now, what I'd like to do next is to look that we have these fissures and sulci that divide the brain into two different main regions. So we have central sulcus. Central sulcus separates kind of the front and the rest of the brain. And we find the central sulcus on all individuals. It's present there. We also have what's known as our lateral sulcus. Lateral sulcus separates this little lobe that's on the side, kind of roughly where your ear would be. And then we have also on the back side our parieto-occipital sulcus and also our transverse cerebral fissure. So these are important because they allow us to separate different regions of the brain. And let's have a look at these lobes of the brain and what's associated with each one. One thing you will notice very quickly that all the work that you put in to learn the different bones that make up the skull is paying off now because these lobes are named after the bones, or bones are named after the lobes that we have on that area. So let's have a look at those. Our frontal lobe is the most anterior portion. It's the front portion. It is the largest in humans, in our evolutionary relatives. It's much smaller, and it is within the frontal lobe where we see many things that are unique to humans being housed, things like thinking, planning, executive functions, and so on. So, for example, the sheep brain that we'll have a look of a video of that got dissected, we'll see that sheep don't do great planning of the future or having big individual personalities. That's because they don't have prominent frontal lobe. We'll also see at the very back of the frontal lobe our primary motor area, kind of a sausage-looking structure where different parts of your body are represented in terms of the motor functions that they do. Then we have our parietal lobe. Your parietal lobe is especially important for sensory functions. We have our primary somatosensory area where entire body is represented, but also other processing, secondary and further processing of sensory information. At the back of the brain, we have our occipital lobe where we do visual processing. I often tell my students the example that when you look at cartoons and someone hits their back of the head, they see stars. Well, that's literally because their visual area has been now physically impacted, so you start to see visual things that don't actually exist. Finally, we have on the side our temporal lobe, and it's within the temporal lobe where we're housing things like language processing for hearing and speech and so on. There are other areas to the brain as well. The two that I've highlighted here, we see our Broca's area, which is responsible for speech production, and Wernicke's area for speech comprehension. Again, a lot of injury studies that have helped us to locate these. But remember, on certain individuals, if you had, for example, damage on your temporal lobe, we can relocate some of those functions elsewhere. Another thing that I kind of wanted to mention is that sometimes we hear people saying that there's a difference between your right and left brain. What we see is that we're talking about a concept known as lateralization of cortical functions. So right hemisphere is often said to house things like your language, your math skills, your logic, whereas left side is more focused on things like your visual, spatial processing, intuition, emotion, artistic, and musical abilities. There is strong views in favor and against of the lateralization of cortical functions. I'm not going to tell you what you should think or what you should believe, but I will say that it hasn't been proven undisputably. So there's still some debate. But some of the belief that 90% of the humans are left dominant for language, these would be our right-handed people, and then 10% is the opposite for us. So you do what you want. And remember that both sides of the brain still do communicate with each other significantly. So it's not quite as simple as these sometimes simplifications tend to make you think that we do have, for example, the corpus callosum connecting the two sides. The next part that I wanted to talk about was our diencephalon. And within diencephalon, we see few different parts that were kind of worthwhile for us to explore. And let's see again if I can take the diencephalon out. Diencephalon is housed deep within the structures of the cerebral hemisphere. There's few parts of those that I want to highlight. So we see thalamus, which is really prominent, large, kind of an egg-shaped blob on both sides. And thalamus is responsible for really fibers that communicate with the cerebral cortex. It's also an important relay station for information coming into the cortex. So it sorts and edits and relays it. A lot of functions like emotions, your visceral functions, some very direct motor functions, your memory and sensory integration, so creating those whole experiences take place at this level. We'll also see that we have a structure, and I don't know if it shows on this picture, but it definitely shows, or on this screen, but it definitely shows on the picture that I provided. We have a structure known as hypothalamus. It literally means that it's underneath the thalamus. And hypothalamus is, for example, an important smell relay station, but it's important because it connects to pituitary gland, which is the gland hanging underneath there. And pituitary gland secretes many important hormones. So we often say that hypothalamus is the master gland of your brain because it controls other body parts, it controls other functions. And then we see that pituitary gland releases further hormones. So a bunch of structures there to be aware and for you to know. I'm thinking, what else? Could I say anything else about hypothalamus since we have it out here? Hypothalamus important for maintaining your homeostasis, things like regulating autonomic nervous system responses like blood pressure, heart rate, digestive tract, pupil size. Also physical responses to emotions. So body temperature regulation, for example, controlling sweating and shivering is from there, hunger, feeling full, thirst, so on and so on. I wanted to highlight one more structure from the epithalamus, especially the pineal gland. And I'm thinking, can I see a pineal gland nicely here? Probably better if I had the brain fully put together so we can use this side of the brain. Pineal gland is this tiny, tiny blob located there, and you'll see it very strikingly when we end up doing the dissection of the sheep brain. And pineal gland is responsible for secreting melatonin. Melatonin is the hormone that regulates your sleep-wake cycle, so it's very important in that sense. What else can we see? Oh, I'm jumping around. We can see our brainstem. So our brainstem has two important portions to it that I want us to know, really three main parts. We have our midbrain. Our midbrain is kind of inside the brain. Then we have the first blob that you'll see. This is known as our pons, and then we have the second, which is known as our medulla, or medulla oblongata, if you wish to use in proper language. And these are like spinal cord, but house vital functions, so essential for our survival. So midbrain is an important relay station. We'll see that pons does things like regulating our breathing rhythm, medulla, things like relay station playing role in coordinating our reflexes, heart rate, also regulating blood vessel diameter often, things like that. Can I get the brain back together? I'll end up fighting with it later on once our class is over. We see that also the regulation of the breathing took place at the level of the medulla. Some other functions that are kind of interesting sometimes for students is that we see, for example, regulating hiccups, regulating things like vomiting, swallowing, coughing, sneezing. So what I give as a general rule of thumb to my students is that the most vital functions that keep you alive are housed within the brainstem. Your heart rate, your breathing rhythm, and so on. That was the first part of your brain to develop in evolutionary sense, but also first part of your brain to develop when you were first developing as an individual. This is also the most protected part, not only by the brain around it, but by the bony structures. So we can lose a lot of cortex, but we want to maintain those vital functions that are housed there. The final part that we're going to have a look of is our cerebellum. Cerebellum, little brain, represents about 11% of the brain mass, responsible for coordinating movements and balance. If we cut cerebellum in half, we'll see a structure known as arbor vitae, which translates from Latin literally tree of life, that is a worthwhile structure. They're important myelinated axons passing messages, coordinating those messages. And I think we'll leave the discussion of the spinal cord to your subsequent chapters, and we'll wrap up at this point. I did want to show, because I mentioned that, regarding the action potential, the question that we were discussing when our class started, and let's see if I haven't been kicked out because there's a time delay. Your quiz 12 on there, question 2 asks you to identify different stages of the action potential, and you'll see that there's a little bit of an oddity that we didn't see and that I really wouldn't associate with typical action potential, which is this part A. So don't let that trip you out. When we're talking about resting potential on this diagram, that would probably be the stage E, where we saw the negative 70 millivolts. That's the resting membrane potential. A in this point would be between when the stimulus has happened and before the actual depolarization starts taking place. B would be our depolarization. I wouldn't assume that we would see stage A in most neurons. We will see that, for example, when we look at, for example, cardiac action potentials, but not on this particular one. I'm just looking, my cat is being naughty and kicking things over. But I do think it's a question that I probably will manually grade or make sure that everyone has a chance to get good grades for that. But since it's there, I'm sharing the answers. Hopefully that helps you a little bit. Don't worry, I would not ask that in an exam. I think it's kind of minute details and details that are not quite what I want us to focus, or even quite accurate in context that we've been talking about the action potentials. So that brings us to the very end of everything that I had planned to share with you. If you do have any questions, I'm super happy to answer those. Otherwise, that's it for this session. I appreciate all 3 of you for joining once again. I hope you get a chance to enjoy your 4th of July celebrations if you participate on those and enjoy of those. And next week, we will be still interacting, even though we don't have our live online session. But stay on lookout on a message about that at the beginning of the week. Thank you again for joining me, and have a good rest of the day. Yes.
Thank you.
Thank you. I appreciate you.
Bye.
Bye. And I'm going to stop the recording at this time. Again, thank you also for everyone. Thank you on the chat as well. Thank you to everyone who joined, and whether it was in person or whether it was through a recording. And remember, I'm only a message away, even though this week I've been a little slow to respond. Have a good weekend. Bye.